I am Raghavi, Assistant Professor, Department of Biochemistry in JJ College of Arts and Science. Now we discuss about a pathway known as glycolysis. In glycolysis is the first step in the breakdown of glucose to extract the energy from cellular metabolism. Glycolysis consisting of an energy requiring phase followed by the energy releasing phase. There are two phases known as energy requiring phase. When we give the energy, it releases the energy in the end. Glycolysis is a series of reactions that extracts the energy from the glucose by splitting it into 2-3 carbon molecules called as pyruvates. The sequence of reaction converting the glucose glycogen to pyruvate lactate with the production of ATPs. This pathway was discovered by Embden Meyerhof pathway, and it is also known as a Meyerhof pathway. Glycolysis takes place in the cytosol of the cell, and it can be broken into three main phases, that is energy requiring phase, splitting phase, and energy releasing phase. First we discuss on the energy requiring phase. In this phase, the starting molecule of glucose get rearranged and two phosphate groups are attached to it. The phosphate groups makes the modified sugar now called as fructose 1,6-bisphosphate unstable, allowing it to split in off and forms the 2-phosphate bearing 3 carbon sugar. That is simply it is gives we give 1 ATP and 2 molecules get up used. In splitting phase, this is the second phase. In this phase, fructose 1,6-diphosphate get dissociated to get the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and final step is the energy releasing phase. In this step, each 3 carbon sugar is converted into another 3 carbon molecules known as pyruvate. Through the series of reaction, in this reaction 2 ATP molecules and 1 NADH molecules are made. This is the following reaction of the energy requiring phase, splitting phase, and energy generating phase. In this phase, glucose is converted into glucose 6-phosphate by a key enzyme known as exokinase, in which we give the ATP and it reduces into adenine diphosphate, and glucose 6-phosphate is then converted into fructose 6-phosphate by a key enzyme known as phosphoglucose isomerase. This is a non-energy requiring phase, and fructose 6-phosphate is then isomerized into a fructose 1,6-bisphosphate with a key enzyme known as a phosphofructokinase. In this step of isomerization, it requires an energy which is given in the form of ATP, and that it reduces into an ADP. In fructose 1,6-bisphosphate, that is a splitting up phases occurs, that is glyceraldehyde 3-phosphate is converted into dihydroxyacetone phosphate, that is converted into 1,3-bisphosphoglycerate with a key enzyme known as GLAP hydrogenase, that is 2 NADP plus 2 PI is converted into 2 NADPH, that is 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate, that is 2 ATP is converted into 2 ATP with a key enzyme known as phosphoglycerate kinase. 3-phosphoglycerate is converted into 2-phosphoglycerate with a key enzyme known as phosphoglycerate mutase. 2-phosphoglycerate is then converted into a phosphoenol pyruvate with a key enzyme known as an enolase. Phosphoenol pyruvate is then finally converted into a pyruvate with a key enzyme known as a pyruvate kinase. In this step of the reaction, we get 2 ATPs, that is adenine triphosphate. This is a complete flow chart of glycolysis. This is a chemical structure of glycolysis. Glucose is converted into glucose 6-phosphate, and fructose 6-phosphate is isomerized to a fructose 1,6-phosphate and splitted into a glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and then it converted into 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, and finally we get a pyruvate from this step of reaction. In the following chart, we know the 10 step of reaction. First step is the a phosphate group is transferred from ATP glucose makes glucose 6 phosphate. Glucose 6 phosphate is more reactive than the glucose, and addition of the phosphate also traps the glucose inside the cell. Since glucose with the phosphate cannot readily cross the members, that is, glucose is the only molecules which passes the blood-brain barrier. In second step of the reaction, glucose 6 phosphate is converted into its isomers fructose 6 phosphate. In third step of reaction, a phosphate group is transferred from ATP to fructose 6 phosphate and produces fructose 1,6 phosphate. This step is catalyzed by an enzyme known as phosphofructokinase, which can be regulated to speed up or slow down the glycolysis pathway. In fourth step of the glycolysis, fructose 1,6 bisphosphate split to form the 2,3 carbon sugar, that is dihydroxy acetone phosphate and glyceraldehyde 3 phosphate. They are isomers of each other, but only one glycerol 3 phosphate can directly continue through the next type of the glycolysis, that is DHAP is converted into glyceraldehyde 3 phosphate. The two molecules exist in the equilibrium, but the equilibrium is pulled strongly downwards. In the schematic diagram shows an glyceraldehyde 3 phosphate used up. In sixth step of reaction, the two half reaction occurs simultaneously, that is the glyceraldehyde 3 phosphate is oxidizes and NAD plus is reduced to NADH and H plus. The overall reaction is an exogenic, releasing the energy that is then used to phosphorylate the molecules, forming 1,3 bisphosphoglycerate. In seventh step of the reaction, 1,3 bisphosphoglycerate donates one of its phosphate group to the ATP and making the molecules of ATP starts and turn into an 3 phosphoglycerate in the process. In eighth step of the reaction, the 3 phosphoglycerate is converted into its isomer, that is 2 phosphoglycerate. In ninth step of the reaction, 2 phosphoglycerate loses a molecule of water, becoming an phosphoenol pyruvate, that is PEP. PEP is an unstable molecule which is immediately converted into a final step of the glycolysis, that is the 10th step. PEP is readily donates its phosphate group to ATP, making it a second molecule of ATP as it lost its phosphate group from the PEP. This is converted into a pyruvate. The end of the glycolysis, we get the ATP. First we must donate the ATP, and final the result of glycolysis we get the ATP. Thank you.